Uh, Good morning, everyone. Can you hear me? Let's see if I can share the screen like this. Yes, okay. Thank you, Itai, for inviting me, and I'm excited uh, to present this uh, exciting topic as well. And let me put my timer. Um, like I said, if you have questions, you can ask during the talk. I don't mind answering questions as long as it's related to the subject, and I stand in my 40 minutes allowance. So today I'm going to talk to you about early detection of soil-borne diseases in field crops using remote sensing, and my partners for this research are Dr. Mary Daphne Yelin, that is a phytopatholog, and also Ophir Dagani, both of them researchers in Migal and Mop Tzafon, dealing with soil-borne diseases. So what are soil-borne diseases? Usually funguses that attack the crops. I am assuming most of you familiar with this more than me. I do not come from the plant science arena. My background is in computer science and hydrology and remote sensing. So uh, my ability to explain exactly the mechanisms of the diseases are inferior to my partners that are experts in this field. But from what I know about these diseases, that they pose significant threat to crop yield because they can attack a field, and within days or even, I mean, weeks or days, they can totally wipe out the whole area. And that's why most treatments are given as early prevention before the onset of visual symptoms. So that's why it's very important to detect these diseases early in order to both reduce the number of treatments and to target the areas that are sick and give treatment only to these areas instead of giving the whole field those poisons that are not so great for the environment and are expensive. And better treatments will prevent the spread of the disease. If we know where the hotspots of these diseases are and we can target them, then we probably could increase the yield while reducing the amount of material. So late wilt is a soil fungus that's called Magna Porthiopsis maydis. I think the name changed not long ago. It's been common in Upper Galilee for the last 40 years. I think it was first discovered in Egypt and spread in this area of the world. And as you can see on the bottom left, in some cases, it can wipe out the whole field. It is usually occurs, you can see it only at the later stages of the cropping. That's why it's called late wilt. If we're talking about 80 to 90, maybe 100 days with the corn for animals, for animal feed, it attacks usually at day 60. Day 50, you can start seeing the first signs of dehydration. So what happens, it attacks the stem, and it blocks the nutrient flow, and it has symptoms like plant dehydration. Okay, and white mold caused by the fungus Sclerotium rolfsi. This is a multi-surrogate disease, whereas the late wilt attacks mostly corn. White mold can attack peanuts, carrots, tomatoes, watermelon, and mainly in the summer, it likes heat and humidity. It has white structure at the root, at the base of the root, and it also makes the plant wilt from lack of nutrients, and also looks a bit like dehydration symptoms. So it spreads in growing spots. Like one area is damaged, then the disease progresses to neighboring plants with the contacts of the organs subterranean. And what we see is a brown, wilted, dead plant. Okay. Thermal imaging. Why do we use thermal imaging? Because the onset of the disease is very similar to dehydration, and thermal imaging can detect water stress and also stress in general, as I will explain shortly. Because every body emits long-wave radiation depending on its temperature, then with remote sensing in the range of 7 to 14 micrometers, we can calculate plants' temperature. And plants in water stress are warmer than plants that are not stressed. Why? Because a healthy plant will evaporate water in the process of photosynthesis, right? And this process also has a cooling effect. Now, when there is not enough water for the plant will close its stomatas, and therefore there will be a decrease in the respiration rate, which will increase the heat balance and temperature of the plant. So this is how we sense stress using thermal imaging. Another technique that we use is visible light imagery. Okay, this is the cheapest way to get data. You can use your phone. You can use any regular camera, RGB we call it, red, green, blue. A visible light image has 3 bands: the red, green, and blue. And we use it on board drones, as I'll demonstrate, as I'll show in a sec. So what happens in the visible range, the GRVI index, which is based on the green and the red radiation, we use it to detect visible stress. Okay, the red light and the green light, as you can see in the graph, there is a difference between the reflectance of the plant. Let me explain what reflectance is. The sun emits electromagnetic radiation in all the spectrum, and most of the radiation that reaches Earth is in the visible range. Now, plants and other objects take some of that energy, and some of it they don't take. They either reflect or transmit. And the amount of energy that is reflected back, this is what we measure with our sensors. Now, a plant needs most of the green light, most of the red light, and the green light, it reflects back. The graph here is a bit, the arrows should move. The red is here and the green is here. And because the chlorophyll, it doesn't use the green light, it reflects it back, and those differences are shown in the GRVI plant index. So a plant in stress will have a lower GRVI value than a healthy plant, okay? Because a healthy, vital plant will perform the respiration and photosynthesis in a better way. So what our research goals are? First of all, was to develop a method for the early detection of soil diseases like the late wilt and the white mold, using thermal and multispectral imagery. Early, why early? I explained. And other than just revealing those hotspots, we also wanted to predict the rate of disease spread and forecast the pattern of spread, where it goes. This part is still ongoing. I'm not going to show much results on this today. The third goal was to explore the potential of large-scale detection of these diseases using satellite imagery. So what we did, we use UAVs, unmanned aerial vehicles or drones, with sensors on board in the visible and thermal spectrum. We compare those different dates because we come on a periodical basis once or twice, once a week or once every 2 weeks, more or less. So we have a very, I would say, high temporal resolution, and we can see the progress of the disease in the field, and we can then compare different dates and see the changes and detect the hotspots and try to make sense of this spread pattern. Okay, we of course validated these findings with ground monitoring that were performed by a skilled inspector, and comparing the timing of finding between the in situ monitoring and what our sensors did in order to see if we can really find or detect the disease earlier than an inspector would do in the field, earlier than can be seen by the visible naked eye. Okay, so this is our Matrix 600 Pro, and we use this one and Phantom 4. Phantom 4 has a built-in RGB camera, and the altitudes we performed the imagery were between 20 to 100 meters. Why 20 meters is because we wanted to get a very high spatial resolution. We're talking about sub-centimeter spatial resolution in order to detect the symptoms of the disease as early as possible. So the Matrix 600 Pro, we had 3 cameras. We had the Sony Alpha, which is a very good and strong camera with very high ISO and stable and good zoom. Here we're talking about 0.2 centimeters. We used AgroWing multispectral lens with 12 channels between 400 and 850 nanometers. And we used a FLIR thermal camera with pixel size between 2.5 to 7 centimeters. And we use Pix4D software images, Pix4D capture for the autonomous piloting of the UAV, creating the flight plan, and Pix4D mapper in order to georeference and mosaic all the images together. Any questions so far? All right. We did about 50 flight missions during the 2019 year, and in 2020 we did around the same amount. I forgot to mention this project is funded by the chief scientist of Ministry of Ministry of Agriculture, and we finished the second year when we are about to start the third and final year of the study, and I will present mainly results from the first year, some results from the second year. We're still crunching and analyzing the multiple amount of data that we have from the second year. And as you can see, we did 50 flight missions in 13 different dates in 4 different sites with the sensors that I presented. Gadash experimental farm, which is the experimental farm of the Hula Valley. We had both peanuts and tomatoes and corn experiments running there. I'm not going to go exactly in detail of the experiments themselves, but what we did is we used different chemical and biological treatments in order to check corn, peanuts, and tomatoes in different stages and severities of disease in order for us to get a range of disease that is presented in the field. And we also use the different varieties, some that are more sensitive to the diseases and some that are less, again, for creating those differences so we can train our sensors. And then we had 3 commercial fields. In by Gonen, we had industrial tomatoes. In Kfar Anasi, we had peanuts, and in Yavne, we had corn. Okay. So, in order to compare the results from different dates and different sensors, and to determine disease hotspots, we had to anchor the product maps exactly one on top of the other, because a slight movement in the position will create a situation that we have different plants in different layers, and we cannot really compare them because one plant could be healthy and the other is sick, and then we get different results from different sensors and also from our ground monitoring. So it was very crucial to do that, but it was difficult to anchor those maps in centimeter resolution because Pix4D Mapper, it is a good software, but it has some distortions that were preventing us from making it really accurate. So in order to overcome this problem, we had 2 different solutions. In small fields, like in the Gadash experimental farm, where we had small plots, we disassembled the mosaic into small parts of 8 by 8 meters. So let's say we had 200 by 50 meters. Imagine the amount of work, the lab and my research associate a lot of work of putting it one on top of the other, all the dates from the different sensors, 8 by 8 meters, and accurately anchoring each part separately. Okay, we had the ground control points that we had the specific location using a handheld RTK GPS. And it was a very hard work, but was necessary for our algorithms. Another solution that we did in big commercial, large commercial plots, we couldn't do this small like we did with the Gadash farm. We did pixel averaging in squares of 25 by 25 centimeters. So we actually reduced our spatial resolution in order to compare the different sensors and compare the sensors to the ground validation. Now, what was good about it is that when we got a big range in our vegetation indices, it meant that there was a big difference within the same pixels, usually happening at the edges, at the edge between soil and plant. We could detect mixed pixels easily and avoid using them in our algorithms. A few more words about thermal calibration. We used a mobile meteorological station that you can see in this picture here. We took it to the field and we measured air, wind, and radiation parameters. And we also had wet and dry references that we took their body temperatures with infrared sensor. We're talking about Campbell, IRTSP, Campbell scientific sensors. And we measured the difference between these sensors and the camera and made sure that there wasn't a big difference between them. Also, pre and post flight, we used the black body to calibrate our camera. We used the SR800 that is created by CA Systems. And I mean, why did we use wet and dry references? Has to do with other vegetation indices that we also worked on, like CWSI. I'm not going to get into this today, but it was a way for us to make sure that the camera is actually working, the thermal camera is working properly. So in order to classify stress using the thermal sensor, we built a classification of 6 groups. Very healthy, healthy, less healthy, stressed, and severely slash dead plant, and soil. Okay, and we took the stressed and severely stressed as plants that were suspected in having the soil disease. Okay, so first of all, pixels that had air temperature, their temperature was higher than air temperature plus 10 Celsius degrees, were classified as soil, and the 15 coldest pixels in the image were classified as very healthy plant. Now, the 4 remaining groups were divided into equal temperature intervals between those 2 thresholds. So in order to validate our classification, we need to have field monitoring at the plant resolution in plot scale. Usually, what we do in remote sensing is that we use something that's called confusion matrix. I will present it shortly. And we take 15% of the field for validation. And for that, you need really good in situ classification, which we didn't have. We're talking about big plots with lots of plants, and each plant needs to be monitored separately. So what we did instead, we used our RGB images as the base image for the classification validation. And for that, we used 2 techniques. One is a person set, and every plant classified it according to its RGB image. And because it's hard to distinguish between a dead plant and soil using RGB vegetation indices, we had to do it manually. Now, another thing that we did was we used 2 additional plant indices that proved to be successful together. We used the GLI and the VARI index, where the GLI index was used to filter the background from the image, filter the soil out of the plants, and then the VARI index was used to classify between a living and a stressed or dead plant. Now, here is an image of that classification validation that we did. I hope you can see it with in your screen. So this is the field in Gadash farm. It's a peanut field, and this is the area that we did the validation. Here you can see it zoomed in. This image is the combination of the GLI and VARI that you can see. White is soil, green is healthy plant, and red is stressed plant. Now you can still see noises, right? The red this thing shows us the stressed plant. So usually a plant is round, okay? This is a stressed plant, but we also have some noises that are in the edges between soil and the plant, and we found a way to get rid of them. But here in this image, I wanted to show you that it's not perfect still, but you can distinguish between the thin streaks in red that are noise and the stressed plants. Image D is the VARI classification by itself, and then E is the GRVI. This is what we used to compare C and E for our validation. F is the FLIR, the thermal image, classified into stress, like I explained before, into 6 different classes. And G is the GLI by itself, okay? So GRVI by itself was better than GLI or VARI by themselves. We use them to create the base map for validation. So this is the confusion matrix. What it is basically what, on one hand, what the remotely sensed sensors saw, and on the other hand, what the manual classification or in situ the reality is. Okay, you usually have a condition positive, a condition negative, and this is an agreement, right? If I saw, like the remotely sensed sensor saw something that's positive, and it's also by the manual classification showed as positive, then we agree this is a true positive. The same with true negative. And these two are either false positive or false negative. Then from that confusion matrix, we can calculate overall accuracy, which is measuring the ratio between the correct identification and the total population. And then there's also something that's called user accuracy and producer's accuracy. User's accuracy is actually to see whether an object marked on the map will actually be present in the field in reality. Okay, so let's say we have a map of our remotely sensed findings. Now I take this map and I go to the field where I told you there is a stressed plant. If you find it in the field, then the user accuracy is higher. Now the producer's accuracy is the other way around, is the probability that the object that you find in the field will be correctly classified on the map. So I think for us, user's accuracy is more important because we make a map and we give it to the farmer, and the farmer needs to go to the field and take an action there. Okay, so if we look at the results of our GRVI findings, we see that overall accuracy is 94%. Kappa coefficient is also very high. I didn't explain. Kappa coefficient is to show that what the classification that we performed is better than just randomly assigning values. Okay, the higher it is, the better. It means that there is a correlation between the findings and the reality. Okay, and user's accuracy is also very high for a plant. For that plant, you see it's a little lower. It's only 75%, and we know we have a problem with it, and we're still working on it. But to distinguish between plant and soil and healthy plant, we have a high agreement. Okay, another thing, we gently, seamlessly jumped into the results. So I want to show you a comparison we did between Venus satellite and our UAV classification. Venus satellites, for whoever doesn't know, it's an Israeli-French satellite for research. run by Ben Gurion University in Israel, and it has a very good spatial, considering other free available satellites, and temporal resolutions. We get an image every 2 days, and that's the temporal resolution, and the spatial resolution is 5 meters per pixel, and even better today when the satellites changed its altitude and it's lower down. So the bottom line here is what we can see with satellite. The middle line is the GRVI image that is calculated from the top line, which is the drone RGB image. So what you see here in this rectangle is an area that is highly, how do you say, sick, highly infected area. These are the dates. So you can see this is end of August, beginning of September, mid-September, at end of September. And you can see that here with the GRVI, the vegetation index, you can see it much better. You can see the yellow and red. These are the hotspots of the disease, and you can see how it deteriorates. Now, with the satellite, you can hardly see any differences. When you look at the mean values of the pixels within the rectangle, so here you see that, you look at the range. This is the satellite, and you can see that the ranges between the minimum and maximum is very low, meaning it's very homogeneous. The satellite did not detect the disease because it doesn't show a difference between healthy plants and sick plants. Whereas with the drone, you can see that the range is much higher, okay? And it is getting higher as the time goes by. Or the onset of the disease is higher. So what we saw is that we could not really detect the disease with satellite, which is understandable because 5 meter pixel, with all due respect, is still not enough to show hotspots or a start of infected plants in a commercial field, at least not at the early stages of the disease. These are results from experiment we did with industrial tomatoes at a commercial field. The graph here shows you the progress of the disease over time. This is beginning of June 2019, and here this is August. And the orange is the infection rate, starting at 20 and progressing to 30, almost 35% of the area that we checked within the commercial field. And the cover the vegetation cover is rapidly growing because of the phenological stages during the beginning of the season. The vegetation cover is going high, but as you can see, because of the disease, it is dropping down from 80% down to 60-something%. And the same goes with the stress. And the stress goes down, and here it goes up. So you can see that there is a correlation between stress and vegetation cover, and also to the infection rate. Okay, and the other graphs show the relationship between stress, the RGB stress, meaning the stress that was measured by the RGB sensor, and the infection rate. At the beginning of the season, we can see that the correlation is high. It's around 75% here. And then it goes down to 60-something and to 50-something. So at the beginning of the season, we had better correlation between infection rate and stress rate that we measured. Also, at the end of the season, it was okay in the 60s, but not that amazing. Another experiment we did with late wilt in Yavne, it was variety check. אני באמצע, sorry. And we measured 10 different species, or 12, sorry, different species of corn. Some were more sensitive to late wilt, while others are resistant. And each, we had 10 blocks, and each block we had all those 12 varieties. And as you can see, this is a close-up of block 4. In 2 different dates, you can see the difference between a stressed and a healthy plant. Let's see the whole image. So red, this is the GRVI, the middle line. Red and yellow is stress, while green is healthy. And the bottom is the thermal imaging. Light blue is hot, dark blue is cold, and hot means stressed. So you can see, first of all, that there is a correlation between the GRVI and the thermal indices, and it correlates to what the findings in situ, meaning the checks that they did on those varieties, and the sensitive ones were more stressed. And you can also see it with the samples that they took. They did some genetic testing, and they found the presence of the disease. Okay, so when we did the overall accuracy for the thermal sensor, we got 93%. Kappa coefficient was lower. Similar results we got for the GRVI sensor. Now these findings are already in review now for publishing, and I hope I will be able to show a journal published paper on this soon. Next, I want to show you results from the experiment we did in a commercial field in Kfar Hanassi with peanuts. What we did was, first of all, we wanted to validate our remote sensing versus the ground sampling and validation. So within the commercial field, we took 8 rows. Each row is 2 meters by 30 meters length. And we had an inspector going every week or so. We divided these each plot into squares of meter by meter. So for every meter, the inspector checked the severity of the infectious diseases. Here we're talking about Rolfs. And we did our algorithm with 2 different thresholds. One was 2.5 degrees Celsius between each group, and the other was 3.5 degrees Celsius difference. So the differences in the degrees changes the sensitivity of our alerts. Okay, 2.5 is more sensitive, while 3 degrees difference is less sensitive, meaning in order to get to the stress, you need to go up 3 Celsius degrees each group. So what's interesting is the difference between hit and miss. Okay, let me explain shortly what we did with this rating. In every area where the inspector said there is a disease, and we said, yes, we see a disease, then we agree that this area is sick. Okay, and areas where we didn't find a disease and the inspector didn't find a disease, we both agreed that it is healthy. In areas where we spotted a disease before the inspector did, that was considered as hit. And in areas where the inspector saw a disease before we saw it, then it was a miss, okay? Now, if you look at the 2.5 difference, this is the monitoring compared to RGB ratio and monitoring compared to FLIR, so compared to the thermal. Both of the, in both times, the field was mostly healthy. So 77% or 79% of the times, we both agreed it was healthy. And around 4 or 5%, we both agreed it was sick. But look at this. In 11% of the times, we said it was sick before the inspector saw it. So we had an early detection 11% of the time out of total 15% sick. And we had a miss of 1%. Okay, now with 3 degrees, because our sensitivity was lower, the hit ratio was lower and the miss ratio was higher. But our false positive here was 0%. We didn't send the farmer to areas that were not sick, claiming they are sick. We didn't have false positives. Whereas with the 2.5, we had between 4 to 6% of the time false positive. So there is a trade-off between the sensitivity of the sensor and the ability to detect earlier a disease while also giving some false alarms. Now, we repeated the same experiment in 2020, exactly the same experiment. Here you can see the squares of 1 meter by 1 meter. The only difference this time is that we chose an area that was more infected. And we started monitoring later where the infection was already starting. So this time we got different results because we both agreed on the sickness. Here, this is the RGB image of the area in Kfar Hanassi. This is the thermal. You can see the range of temperatures between 36 to 62. 60s are, you can see this is the soil. Red areas are sick areas, infected plants. And this is the classification we did. Here we did a bit different classification. I will explain if I have time. And this is the results of the ground in situ by the inspector. Now, when we look at all of them together, we see that number of cells that we agree healthy was 0, meaning there were no, at the end of the season, there were no really healthy plants, okay? Most of them were sick, okay? So we agreed on 60%, 16%, both of them, we agreed that they are sick. But what is significant here, that 65% of the sick plants, of the total plants, we detected the disease earlier than the inspector, which is very significant. We also had 18% false positives and 1% miss. Here, we used a slightly different method to classify. I'm not going to go into this because I don't have too much time. But if you're interested, I can talk about it later. Another thing I wanted to show is using a multispectral camera. This is a RGB image from the multispectral camera. This is a close-up to an area that was more infected. This is the NDVI. Because we have a multispectral, we can also use NDVI vegetation index that uses the near infrared spectrum. And this is GRVI. You don't see much differences between them. And this is something that we found that the 405 and 650 bands can give us a good look at dead plants, okay? When we look at all of them together, you can see that both NDVI and GRVI are fuzzy and they do not detect the difference between soil and dead plants very good, whereas with the 405 band, we can see the difference, that the soil is more red and infected plants are less. There is a difference we can distinguish between them. Here, with GRVI and NDVI, it's harder. So this is something we want to continue working on. One last result I want to show you, I think, is analyzing disease spread. Here we can see both RGB and thermal imaging from different dates. And you can see the areas. Here you can see a start of the disease. Here you can already see it sick, and here too. And you can see the spread of the disease as a function of time as the circles grow. What's interesting is that we could see those, not in this picture, but we could see the areas here that were stressed. We could see it earlier on with the thermal sensor. And also with the thermal sensor, we can see a plant that is stressed even before we can see the changes visually, okay? Now here we also at the beginning of our algorithm process, we're still working on it, but what we have here is qualitatively rather than quantitatively results. So in order to summarize the proposed classification that is looking at the 15% of the coldest pixels and is looking at the higher threshold of air temperature plus 10 degrees Celsius was proved successful in classifying the plant health status and locating disease outbreaks. The thermal sensor was able to detect the onset of the soil disease even before it could be seen by the naked eye. And as I showed, we were able to see the disease with the thermal and also to a certain extent with the RGB sensors before the inspector was able to notice them on site. And we're talking about an inspector that came to the same area every week and looked at those squares of meter by meter and even looked under them to see the Rolfsy white mold thing. In reality, inspector won't go in that detail, but even with that level of detail, we were able to beat the inspector. Now, there is a trade-off between the ability to early detect and with the lower miss levels, you get a high level of false alarm. And this trade-off can be handled. We can change the sensitivity of the model according to demand, let's say. Like I said, it was difficult to distinguish between a dead plant and shade of the plant or soil using the GRVI classification. We got an improved distinction using both the GLI and VARI and using the 405 band. So this is something we're still working on, and I hope we'll be able to produce better results. We had better ability to detect infection. If you remember the graphs talking about the tomatoes at the beginning of the growing season, it's also due to the nature of the disease. Both diseases, or at least the Rolfsy disease, what we saw with tomatoes and peanuts is that we have two periods of high infection. At the beginning of the season, right after sowing, there is a lot of plants dying, and also towards the end of the season. In the middle of the season, not too much plants dying. Like I said, the 405 nanometer channel is a promising channel in terms of detecting dead plants. Satellite imaging was not able to detect the early stages of the disease. And the solution of creating large pixels with average values also proved to be successful in order to detect disease outbreaks and also to distinguish between soil and plants and taking those mixed pixels out of calculation. monitoring of plants over time enables us to detect focal points of disease and to calculate the spread. There are many factors that influence the spread of the disease, like the temperature, the humidity, the amount of irrigation, amount of sun, and other parameters. And I mean, this is why it's hard, but we are starting to being able to detect the patterns of spread, and hopefully we'll be able to give an answer to that at the end of this year. What we're planning to do this year is to use machine learning to enable us to detect and separate dead plants from soil. And we're working on our forecasting model to detect the spread, not just to, one thing is to detect the hotspot. The other thing is to be able to detect the spread and give a forecast of where the disease is going. And we're going to use both also hyperspectral bands to identify the disease. I want to thank you for listening, and sorry for going over time. And if there are any questions, I would be very happy to answer.
Thank you very much, Assaf, for the lecture. It was thorough and on good pace. And first of all, we have a question from Ron Zeligman. Ron, do you want me to ask it? It's in the chat, or you want to do it? I don't see. Okay, so I'll ask it. Why do only the R and G wavelengths used and not comparison of the full spectral signature?
Okay, I see in the chat here something else. Was the VPD measured?
It was sent privately to me.
Okay. Why was the RGB used instead of?
Why do only the RGB wave R and G were used and not comparison of the full spectral signature?
Okay, so first of all, we did not have a hyperspectral camera. What we had was an RGB camera that gave us 3 bands. That's what we mostly used. There are 2 reasons for that. First reason is being practical. No farmer will have the ability to use high-end cameras that cost like $50,000 to $100,000 to analyze it. I mean, getting the whole signature is something that we want to do. Maybe we'll do it this year using the hyperspectral. We just got a new hyperspectral camera, will enable us to do that. So far, we didn't have the tools to do that. And we were trying to use RGB for practical reasons. If we are able to find the onset of the disease using only 3 bands, that's a bonanza. And then every farmer that has a simple Phantom with a built-in RGB camera that costs like $1,000 can get that drone and take an image of their field, send it to us, and we can, or whatever, with an application or whatever, they can find out the disease spread or at least the hotspots. If that's not possible, then you need somebody else to do it for you. I agree with you that it's much better to have more data, but sometimes it's more confusing and sometimes it's not needed. And we showed that we're able to detect the disease with only 2 bands, the green and the red. I hope that's answering your question.
Okay, Ron is not talking, but is replying in chat. So if it makes sense, Ron, please let us know. Shiran also wanted to ask a question.
Yes, Shiran.
And then feel free after Shiran, feel free people to ask.
Assaf, you mentioned that often you are able to detect the onset of a disease earlier. Can you say by how much? I mean, And,
Yes.
and also, if you know if the physiologically, if it's still early enough to prevent a bigger spread, and I mean, if it's applicable.
Yes. So
not only, may I add, not only
yes.
to prevent bigger spread, is it early enough to treat and stop the disease completely and
In the sick plant.
ameliorate or prevent the damage?
Okay. I'll answer the first question first because that's something that I can tell better, and the second I can maybe give a partial answer. The first question was how far early enough, how early can we detect it? So in some cases, we're talking about up to a month earlier. So in 2020, we were able to detect stress using the thermal camera a month before, up to a month before it was detected by the inspector. We also have another experiment that not I did, but Mary did. Mary Daphne that is researching the Sclerotium rolfsi. They took plants, they inoculated them, and they started checking from how, like, from inoculation, how long it takes them to actually die, and it takes a month or more. So that shows that up to a month earlier, the detection is valid. Mostly we're talking about a week or two, but up to a month is also possible. Now, to answer your second question, I don't know if a plant that is already sick can get healthy. I'm not an expert in this field. I'm suspecting that not, that the chemicals that they use is preventing the spread. So if we can see the disease 2 weeks in advance before the onset of the visual symptoms, and we can apply the chemical to that spot in 2 weeks before, I am assuming that the plants surrounding the sick plant will stay alive and healthy and give a normal yield. I'm not sure about the specific plant, but I'm assuming this is not as important because the surrounding area is what's important. That you pinpoint the areas of sickness and you give only that area an early preventative.
Certainly, healing the diseased plant, the infected plant is not so important. What is most important is preventing an epidemic in the field.
Yes.
I
So
also,
yes.
no, sorry. How do you know that it's really a disease? I mean, everything you show could easily describe drought, salinity, malnutrition, and so on and so forth. So I don't
Right.
really understand how you so surely define it's a disease.
So in those areas, the results I showed you, we both in the experimental farm in Gadash and also in the commercial fields, we took areas. In the commercial field, we took 8 rows by 30 meters. We're talking about 16 by 30 meters that the inspector, it's an expert detecting the disease. She went every week and she checked every plant in those sections. And if it was aspergillus, she said it was aspergillus. If it was rolfsi, she said it was rolfsi. If it wasn't a disease or it was just bad irrigation, then we know it was something else. So we had an idea of what's in the field. That was the how would I say, the in situ validation I was talking about.
Can I just
No,
add?
but I'm talking about
Yes.
the future perspectives, because what you show is, of course, validation, which is clear for your particular result. No argument. It's beautiful. But if you want to take it for the future, for other fields, when you don't have agronomists that can go every week to check meter by meter.
Yes.
So how can we take your result and implement them on, and of course on non-irrigated field, which are the majority in the world, and then you really have other background issues that the question is, how can you filter them?
That's why we were taking our time. We have 3 years of research that we are actually trying to pinpoint the disease symptoms and separate it from other symptoms. For sure, there might be other diseases, but the purpose of this research is to be able to pinpoint those specific to soil-borne diseases. That's why we have the validation, and the results are relevant to specific detection of these diseases. I'm not sure I get what you're saying.
No, what I'm saying is very simple. For example, if the field was not well irrigated, you will get the same symptoms exactly. Hotter plants, which will look like you show disease plant. If you have lack of nitrogen, you will see the same symptoms. If you have salinity stress, you will see similar symptoms. That's what I mean. I mean, many abiotic stresses will look the same. So this is my question. How can you separate this soil-borne disease from many other aspects that could look the same?
Yes, okay, I understand what you're saying. We did not check stressing the plants with irrigation, but it's a good idea to do it in an experimental farm where in a commercial field, nobody is going to do it for us. But it's a good idea to have an area that is not well irrigated and try to see the symptoms and the differences. I'm assuming that the area that both doesn't have good irrigation and has the sickness, well, I'm not sure, because the Rothsey likes humidity. So if you starve the plant, you might not get the Rothsey full power, but the plant will die. What basically I'm saying is, you might be right. I might not be able to detect the differences between a wilting plant from water stress or the wilting plant because of Rothsey or late wilt. But what I'm saying is, okay, I don't care. The plant is suffering. You need to go and check. I'm going to give you the areas where I think there is a problem. It could be an irrigation problem, or it could be a sickness problem. You go there and you see in your eyes what it is and give the treatment. Okay, it might be another abiotic stress, but at least I was able to show you an area where it needs attention.
Okay, thank you.
Just to widen this thing, so I had this question for Menachem 4 years ago, a little bit less. And if I remember correctly, what I answered was that I think it takes a combination of few more things. Like you said, it takes, like Assaf said, it takes not only the disease itself, but you have some prior knowledge because you have maps from previous years, you know if your irrigation is working or not, and etc. It doesn't say it will answer the question. It will not give a perfect answer, but it might be improving. And as Assaf said, anomality is easy to detect. And then go and look. This is what I have to comment about that. Are there any more questions?
I see is there a time in the day irrigation cycle better for detection? Oh, I see in the chat here a question.
It was sent privately to you, probably.
Is there a timing in the day irrigation cycle better for detection? If I missed it in the presentation, apologies. Okay. No, you didn't miss it, and I forgot to mention it, that where the timing of the day where we take the images is midday, when the sun is above the field, full on, and the stress is maximal. In the summer, this is the timing of the day where we usually take our images. We also prevent that, with that we prevent other problems like how do you say, atmospheric correction and so forth, and BRDF and other problems that has to do more with the physical aspects of the angle of the sun. I hope I answered your question.
I think we are already past 10, so If there is any urgent question, please ask now. If it can wait, I'm sure that Assaf will be happy to get comments and communication. So if there is any urgent question, it's last chance now. Okay, so if not, so thank you very much. Have a good week. Next week, Doron will present Aaron Fiat from Sde Boker, and have a good one. Thanks. Thank you, Assaf, very much.
Thank you for listening, and I'm available. My email, I forgot to mention the great help of all the people that helped in this research, and also the farmers, both Ofer Blau and Eran Kainan, that were kind enough to give their fields for our experiments and cooperated great. And thank Mary, and thank Ophir, and thank also Moran and Gil, my research assistants, and also the research assistants of Ophir and Mary, Shlomit Dor, and Dekel Avraham, and Niva Shaked, and whoever I forgot. On Rabinovich, of course, is also a partner. Thank you. Bye-bye.
Thank you.
Thank you. Have a good week. Bye-bye. Yeah. Do you want to stop?